as well. So let me end the poll and share the results. So Orbitrap is the winner, but there's also some Bruker and
It's nice
others.
to see. It's nice to see that there is a diversity of instruments used.
That's right. Okay, so this is all the polls that we have for you. So now, Alexey, it's your turn.
All right. So thank you so much again for inviting us. Um, and for introducing me. And I don't need to introduce myself again. I would like to really, again, thank my lab and introduce Sarah. She's here in the middle. It's the pre-pandemic photo, as you can imagine, and then Finchawa on the right of the photo, and Danny next to Sarah here. So they will be helping with answering questions as well as with some of the a live FragPipe demonstration later after my seminar, after my presentation. So I'm really grateful to have this lab and it's just terrific to work with them. I also want to mention many collaborators that work with us. I cannot acknowledge them all. I'll mention some of them during the presentation, but I'm always really grateful to collaborators who push us to develop our tools, sort of take them to the next level. It's really important for our research to be driven by high quality collaborations. So FragPipe itself, you will see it later in much more detail. Hopefully some of you will download or already downloaded. Obviously already some of you using it actually. It's really started as a graphical user interface through a MS-Fragger database search engine that we published about 4 years ago, actually, in fact, 4 years ago, almost exactly. So initially it was a very limited scope. We wanted to develop FragPipe as or rather MS-Fragger GUI as a graphical user interface to this really awesome search engine that's hundreds of times faster than commonly used search engines. And if anyone in this audience would like to know more about why it's so fast and talk about a little bit more about details of database searching, I would be happy to talk to you, ask those questions. We have some time after the presentation and we can discuss that. The main focus of this presentation is going to be on quantitative workflows. So we'll try to get to that as fast as we can. Now, I also should mention that, MS-Fragger is just a search engine. So we had to develop a lot of infrastructure around it for peptide and validation and protein inference, false discovery rate calculations, and PTM annotation, and so on and so forth, right? So we published Philosopher as a versatile toolkit that allows us to do a lot of these additional steps that happen after the database search is completed. So that was the next huge step for us in the process. And once we get this in place and we realize that, we are also working on really new algorithms for label free quantification, that we have DIA workflows and so on and so forth, that we really pushed ourselves to create FragPipe as a complete workflow covering all workflows really. So again, started with a pretty modest graphical user interface and developed into a full-fledged sort of complete computational environment. So right now in FragPipe, we have several key workflows, and you'll see how easy it is to actually select a specific workflow and customize it and run it on your data. It's just most of these things here take just a few clicks. But we have workflows for building spectral libraries for quantitative analysis using DIA. And the libraries can be built from DVA data, from DIA data using DIA and Pyro combined. We'll get back to that at the end. We have LFQ with match between runs workflow using IronQuant that also now supports Scilab data as well, so not only label free. We have TMT quantitative workflow, so iTrack that we run on huge data sets as part of the CPTAC consortium, for example, works really well. And of course, at the core of that, something that's really of great interest to my lab is PTM analysis using open search, massive search strategies enabled by fragment indexing of MS-Fragger. So that certainly is a very popular workflow in FragPipe, sort of MS-Fragger philosopher and PTM shepherd for mass shift annotation. So I'll get back to some of these things a bit later. Now, advantages of FragPipe computational platform compared to other tools. Well, I think it's really one key advantage is that, as I mentioned, open searching and a comprehensive PTM profiling platform that we developed based on open searching concept. That's really applicable to either QC scenarios, but also to finding real biological modifications, RNA protein crosslinks, chemical proteomics. It's really, we pushed it into this area of glycoproteomics and labile modification profiling in general, something that's very interesting to us. It's also great for standard searches that take too long. For example, endogenous peptidomes or HLA peptidomes, any sort of non-specific searching, it takes a lot of time, and it's good because Fragger is fast, but also we optimized a few other things in the pipeline, and of course you can couple it with quantitative analysis as well. Interactome studies, proteogenomics. I don't have time to discuss proteogenomics, but ability to identify this unexpected mass shift is important for that, as well as the speed of the analysis. But I'll focus most in my presentation on the quantitative workflows, as I mentioned, and also a little bit on the spectral library building for DIA. Now, very briefly about MS-Fragger and the PTM analysis. MS-Fragger is a fast index-based search tool, so it enables the concept of open searching that we improved recently by publishing this new algorithm that we call localization-aware open search that solves a lot of initial limitations of open searching. We developed PTM Shepherd and a few other tools on top of open searching so that we can visualize and summarize open search results. And we are working now on coupling open or massive set searching concept with quantitative workflows as well, especially the library-free workflow and LFQ workflow and library building workflow. So this is actually a work in progress because it's a bit challenging, open search and quantification at the same time, but we are working on it. Some of that is already functional. Now about why our interest in open searching as well in general, and that's because I've been personally fascinated by the dark matter of proteomics for a long, long time, asking questions of what are those unassigned high-quality spectra that we have in our data sets. And in fact, this was one of the main reasons we developed MS-Fragger and open searching concept in the first place. So really, MS-Fragger is a search engine that's the dark matter telescope for us, and FragPipe is infrastructure for running it. I already mentioned that. It was published about 4 years ago, and it goes beyond the standard strategies of iterative searching, sequence tagging, dependent peptide search approach, and really implements this open massive set database search approach to PTM analysis. And again, I do not have time to go on this presentation into this localization-aware open search, but I welcome everyone to read this recent paper that we published last August that really takes open searching concept to the next level by showing how we can incorporate fragments containing modification and scoring. Because initially, the straightforward open searching was only based on identifying peptides by unmodified Y in the ions. So really, this algorithm enabled us to do much better with MS-Fragger and open searching in general by incorporating those ions in scoring. So if you're interested, you can please read that manuscript. And to sort of to summarize what we have for open searching as a workflows, in addition to the conventional closed search, that's really where we search for specific peptides that have mass to charge ratio of the peptide matching the observed mass to charge ratio of the experimental spectrum, really looking for unmodified peptides only or peptides containing specified variable modifications and maybe isotope errors. So we go away from the closed search to full open search, where we really search for any mass shift and allowing any mass shift on the peptide. For example, from minus 100 to 100 or from minus 100 to 500 delta. It's a parameter in the tool. And also there is this third strategy that's somewhere in between closed search and open search. And that strategy allows us to specify a list of specific mass shifts that we're interested in. So not any mass shift like an open search, but for example, acetylation and phosphorylation. So we can tell the program to search only for those mass shifts in addition to the unmodified peptides that don't have a mass shift. And that's actually a very powerful workflow for glycoproteomics, for looking for protein RNA or protein DNA adducts, and for a lot of other applications. And if you do a full open search, you will reconstruct all mass shifts that you observed in the data, and common modifications, whether they're biological artifacts, will show up as this in this histogram of PSMs as a function of a delta mass as this peaks at particular values. So, for example, phosphorylation would be peak at 79.96. And so on. So you could use this, as you see from this slide, this strategy is a very obvious sample QC readout, but it's also great for discovering real biological modifications as well. And in general, OpenSearch gives maybe something like 30% increase in the number of identified spectra, 30-40%. So there is a very big boost because we have a very large number of those modified peptides. Granted, a lot of them are chemical modifications and maybe not as interesting, but as I said, a lot of them are biological modifications as well. And by going away from the conventional closed search, you can significantly boost the number of identified MS spectra. So it's probably the most noticeable in the context of APMS studies, where you could take, for example, bioplex data, where it's if you run a particular bait proteins enriched and you're capturing the bait protein and its interacting partners. And what you will see if you do an open search in APMS data set like that, that on average, you're going to have 400% more PSMs identified for bait proteins because they're highly enriched. So this plot shows each dot here the different APMS run from the bioplex 1 data set. And this is, for example, one run where the bait was ETHE1 protein. And what you see here is that on average, in a closed search, we get, for this protein, we got about 50 peptide ions identified in a conventional search. And we got over 4,000 PSMs identified in an open search. And that's because those proteins are highly abundant and some of them are heavily modified post-translationally. In fact, if we look at this particular data set for what proteins gain the most PSMs when we went from closed to open search, we found out that those proteins were enriched for this particular small molecule metabolic processes category in this data set, suggesting that these are the proteins that when they use these baits, these proteins are modifying other proteins by placing small molecules in them, and they modify themselves. So perhaps not surprisingly, proteins that involved in post-translational modification are themselves the most modified, and as a result, we got the highest gain in the number of PSMs identified when we went through open search. So key improvements in MS-Fragger in the last 2 years, in addition to the localization of our open search that I mentioned, is that we introduced MS1 and MS2 mass calibration. We also added search parameter optimization, implemented the isotoping, adapted the algorithms to FAMES data as well as timsTOF data, implemented monoisotope correction, and quite a few other technical improvements that really boosted the performance of already pretty good performance to the next level. If any of you are also interested in really exploring these PTM profiles, I want to highlight this PTM Sheffer tool that we published last year. So Danny will be participating in this presentation. He can answer questions if you have any related to open searching. And this tool will summarize where a particular mass shift is localized, look at the spectral similarity, retention time shifts caused by the modifications, relative abundance changes in modified compared to unmodified peptides, and a few other things. So it's a very useful tool. And you could use this tool to even discover new or unexpected modifications, as we are showing in the manuscript, where we found some unexpected cyclization event on synthetic peptides from Bernard Kuster's synthetic peptide study. Without going into too much detail, again, I invite you to go and explore the paper and see how we illustrate the use of PTM Sheffer on top of open searching for that purpose. You could also use it for summarizing PTM results across multiple experiments. So PTM Sheffer can generate a multi-experimental report where it will compare PTM profiles from different experiments, and you can visualize them as heat maps and see if you have a particular experiment where you have a lot more of adducts due to sample handling, or if there is a biological modifications that for some reason is particularly enriched in one set of experiments, and so on and so forth. So we also illustrate that application in the PTM paper as well. Finally, for this part, if you're interested in the glycoproteomics or lab modifications in general, we published a manuscript in Nature Methods on glycoproteomics. Really, we demonstrate how these strategies enable fast and comprehensive NO and O-linked glycoproteomics. And this work by Ben Pulaski. So it's really an extension of what we've done before, and it's really working great. And we are now working on further improving this strategy by better annotating glycans that we identified with the open search strategy. So let me move to quantitative workflows in FragPipe, which is what I wanted to spend more time on, and really show you that we have a lot of workflows that's already a preset for you in FragPipe. So as a user, when you go to FragPipe, when you started, you do not have to think too much about your setting details of a particular experiment. For example, if you want to build a spectral library using DIA data, using DIA inquiry, all you need to do is just to load DIA spectral library workflow by clicking on load, selecting, clicking on load the data, and really everything that you need is already set for every tab here. So we'll get to that later in a hands-on sort of session. Now, let me move to this quantitative workflows in a little bit more detail by starting with label-free quantification with MS1 quantification with IonQuant. So this work really was enabled by this two recent manuscripts that we published in MCP. They're sort of, the second one is a significant extension on the first one. This work is led by Fin Chao as well as Sarah in the lab. And let me go through the key steps in this IonQuant label-free quantification workflow, and specifically talk about a false discovery rate enabled match between runs that we implemented, that we think is really a key part of any label-free quantification workflow. Now, the advantages of IonQuant in our pipeline in general here, it's again fast because we are using indexing. I briefly mentioned that we used indexing for MSMS database searching. Well, and there we indexed the database, we indexed fragment ions that we generate for peptides in the database. But in a similar way, we can index the data, experimental data, and we can really do peak tracing in multiple dimensions, including ion mobility very fast. So indexing really speeds up a lot of things. We added ion mobility support, so I don't know many tools that really provide, or at least available tools, that provide native support for FAMES data. So we have that. We added a false discovery controlled match between runs. That's very important, as I mentioned. And we even extended it to SciLAC data, because we have a lot of collaborators that really would like to do that. So now SciLAC is also fully supported in FragPipe through IonQuant. So let me tell you about the first part, which is peak tracing and signal processing in general. And this shows for timestamp data, we have an additional ion mobility dimension. But the steps are probably the same ones as we would normally go through, where you would build the index of the raw data. Again, indexing is a key part here. We have MOZ, retention time, ion mobility dimension. So all this data is indexed for very fast and data manipulation later. And we cluster features, we trace peaks in ion mobility retention time, we determine the peak boundaries and calculate the apex of the peak or a peak area. Now we're currently using peak area as a measure of protein abundance, or rather peptide ion abundance. And it's very flexible because we have options in FragPipe in the IonQuant tab that control a lot of the steps. So I think it's very powerful because while we preset the optimal settings, we also give the users the possibility to change the parameters in how that's done. Now let's talk about the match between runs part here, because I think it's pretty interesting as well. So in this match between runs, and of course, most of you are hopefully familiar with the concept of match between runs. It's basically we would like to transfer notifications found in one run to another run where those particular peptides were not identified by MSMS, either because MSMS was not acquired or because the spectrum was not good enough to identify the peptide. So there are key steps here. The first, for each acceptor runs, and the terminology here that we use is that we have an acceptor run. It's the run that's where we would like to transfer notifications from other runs. And those other runs, we refer to them as donor runs. So for each acceptor run, we find n closest donor runs. N is a user-specified parameter where we ranked for each run, for each acceptor run, we ranked all other runs according to the score. That's a sort of a combination of a correlation in retention time, sort of an intensity of peptides identified between the two runs, as well as the total number of notifications that overlap between the two runs. So we create sort of a similarity between the run score, and we rank all donor runs based on that score, and we can allow transfer of notifications from up to n closest donor runs. And for each qualified donor run that we select based on the first step, we align retention time and ion mobility when applicable and transfer missing notifications from those runs into the acceptor run. And in doing so, we also transfer decoys. So that's a really key part that I'll explain later as well. So a few things to think about here is that for each acceptor run, what do we specify as the n closest donor runs that we allow for transfer? Well, in one scenario is, for example, you do a profiling study, and each run is a single shot run of a different sample, and you would like to really allow transfer from every run to every other run. Well, you could set n to the number of runs that higher than the total number of runs in the experiment. However, if you have thousands of runs, obviously it's going to be computationally, I wouldn't say time prohibitive. People have done that actually, but it may not be, it wouldn't be fast, but manageable. But it may not be a good idea just because do you really want to align every run with every other run? I mean, if you know, we now in our experience, we think about 10 runs is sufficient, even if you have hundreds of runs. There is no point of trying to rescue an notification from a run that's not identified. That notification is not identified in 10 other runs that are very similar to the one to which you try to transfer. So I would argue that probably specifying n to something like 10 is probably sufficient even for large scale data sets. Alternatively, one can specify a very large n, but restrict the correlation between the runs that are used as a qualified donor runs. So this is sort of illustrated here. In the middle, we have an acceptor run, and then every other run in the data set is a dot here. And if it's really close to the acceptor run, it means that it's very similar with respect to number of notifications and the correlation and the retention time and ion mobility. And if it's far away, it means that the runs are very, very different. So we could specify a correlation of 0.5, for example, and that will give us this gray circle. So in that case, only runs inside that circle will be used. Or we can specify a correlation of 0.5 and restrict to the best 3 runs. So in that case, these 3 blue runs will be specified as donor runs, and this run, even though it's also similar to the acceptor run based on the correlation score, but it's number 4, it's a bit farther away and it's not used. So we have these 2 parameters to really control what runs are allowed as donor runs here. Now, there are some cases where you may want to allow a particular run to be used as a donor run, even though it's very distinct from the acceptor run. So one good example is when you do a single shot LFQ analysis, you have single shot data on many samples, but you also have fractionated samples that you use as boosting runs, as library runs, if you wish. So those fractionated runs, because each fractionated run is only one particular peptide fraction, maybe a pool of all samples fractionated using SCX or something like this. So each library run would probably be quite different from any other run, any single shot run. So it would, the correlation would be low, but we would like to use that run to transfer notifications. So we can annotate them in FragPipe with a sort of keyword library run. And then everything that's annotated as a library run will be used for match between run transfer, even if it would not pass other criteria such as top n and the minimum correlation that we specify. So Another scenario to think about that's really interesting is we often have a situation when we have two conditions, for example, condition A and B, and say we have three replicates for each condition, and for each condition, maybe for each for a particular protein, like protein A here, in condition A, we identify and quantify this protein in one replicate, in replicate A1, and we don't have any other identifications for this protein, including nothing in condition B. Now, it's very tempting to use this strategy of, like a max quant has a strategy of setting fractions, in which case match between rounds would be allowed only across condition A or across condition B. And we have an equivalent of that. For example, in this case, if we set n equals to 2, obviously other runs from the same condition are going to be closer to each other than two runs from the other condition. So by setting n equals 2, we'll effectively be transferring for A1 only identifications from A2 and A3, and you would prevent transfer from A to B and the other way around, right? Now, it's tempting to do that, but in our experience, it's a dangerous strategy to apply this constraint, because in this example here, if you identify this protein in only one run, it would not come up as differentially expressed protein probably, right? Because we don't have IDs from the other two replicates. But if we set match between runs, we may find it also in A2 and A3, right? But because we do not allow transfer to B, it will stay as NA here. So we found that this strategy creates a lot of falsely consistent differential expression that later, if you're not careful, can give you statistically significant differential expression using approaches that sort of do missing value imputation or just sort of replace things with zero and so on and so forth. So that's really sort of something to think about, and we do not recommend this. I would rather probably always use match between rounds across all, or at least across some rounds between different conditions in this scenario, to avoid this this trap of creating consistent identification across one condition only because we allowed MBR within one condition. So another important part here is false discovery rate, as I mentioned, and we have an LDA training step. Sort of, I don't want to go too much into details now; it's published, but we basically using ions identified in both the acceptor run and in the donor run, we can train, you can compute the scores and train sort of a composite score that then we can use for computing the posterior probabilities for each transfer, for each type 1 ion, which is the ions that are missing in the acceptor run. That's what we're interested in. So we train it on runs on ions that were identified in both runs and apply it to ions that we are transferring. And we use the mixture modeling approach here, where we use decoys to sort of pin the distribution. So we model the target distribution, but we model the negative tail of the target distribution by using the knowledge of the decoy scores. And what are the decoys here? The decoys here are really ions that we create an ion with a shifted MOZ value. So for each donor run, for each ion that we transfer from the donor run to the acceptor run, we also create a decoy ion that's shifted by a certain MOZ. That's, I think, 11 Dalton, that's using the PNNL sort of approach from many years before, developed for sort of AMT mass and tag approach before the conventional strategies in a way. But anyways, we create this decoy transfers and we calculate scores for both target transfers that we're interested in and decoy transfers. And here is the list of scores that we compute. Nothing too surprising, such as retention time difference between a line, retention time difference between the ion and the donor run, and in the donor run here, and the ion and the acceptor run where we transfer, as well as the ion mobility difference and absolute mass deviation in ppm and so on and so forth. So for each donor run, for each ion, we transfer the ion itself, and you create a decoy and search for a decoy here. And if you find one, then we identify a decoy, and we have a score, and hopefully we can control the rates of those decoys by our mixture modeling approach and full discovery rate estimation. And this is from a real run. This is so ion quant will generate these figures. You could see how it works in practice if you use LFQ with ion quant. So the red ones are the transferred ions that we missed in the acceptor run. And so we're modeling this red distribution. And this is the blue is the decoy distribution. And the green one is the fit to this negative tail of the red distribution by using the knowledge of how decoys look. So really, we compute the posterior probability by taking the ratio of the yellow and the green with the distributions for each transferred ion. And using that computed posterior probability, then we can calculate for each scorer a full discovery rate estimate. And then we can go to and apply, say, 1% ion level full discovery rate cutoff. So, and report only those transferred ions that pass this 1% match between run FDR cutoff. And that's what we have implemented by default. So we applied this and to a number of data sets. For example, we applied it to a sort of a pretty standard just replicates of HeLa cell line data, timestamp data for replicates, and we compared it with MaxQuant. And for both MaxQuant and ion quant, our ion quant, we have an option of allowing any protein to be quantified or requiring at least two ratios, two ions for quantification. So of course, applying two ions compared to one ion improves the CV across replicates. So for example, we go from 4% to 3.6% with match between runs, although we do lose some quantified proteins as expected. More importantly here, we compare basically for the same settings, compare ion quant versus max quant, for example, with match between runs and requiring two ions. Max quant identified 5,773 proteins, and we have about 10% more, 5,500, and we have a notably lower CV compared to max quant. In this panel on the right, we are using a 3-organism data set where there is a human, yeast, and E. coli mixed, and E. coli and yeast are, at 2:1 or 1:4 ratios. So blue is yeast, and orange is E. coli, and green in the middle is human. It should be 1:1 between the two conditions. And as you see, max quant on top, ion quant on the bottom, with and without match between runs. So here is with match between runs. Again, we quantify in this data more proteins than max quant, but importantly, we see visibly a lot less outliers compared to max quant in this comparison. So there are a few other comparisons that we've done on the paper that I welcome you to go and see and look really at the accuracy and sensitivity of computing protein ratios, as well as peptide ion ratios and absolute intensities. Now, we also apply it to single cell data set, actually 2 single cell data sets. I think it's really great. These are really great data sets because I think LFQ with MBR is a good strategy for such data. So we use this data set from PNNL, published last year on analytical chemistry with 4 replicates of 50 cells used as boosting samples and 11 replicates of single cell data. So here, this is the single cell data, and there is also some 3-cell data and 10-cell data. So in the single cell data, each dot represents a different single cell, and we have ion quant versus max quant, with and without match between runs. And again, to summarize, we consistently identified more proteins, quantify more proteins than max quant when we use match between runs. Importantly, we can control the match between runs with max quant. For example, you just don't know what kind of full discovery rate for MBR you have, where we can control it at 1% or 1%, so we actually know what the numbers are. So here we're showing the green, for example, is 1% MBR versus max quant is unknown error rate. And if you looked at CV values, they're also in ion quant with match between runs. This plot at the bottom shows the number of quantified proteins in total for the single cell data, or rather, right. And also what's the CV across the replicates of single cell. And it's comparable or slightly less than max quant. So I think we can say that we really get a comparable performance to max quant, if not better. based on all this analysis. We have more proteins quantified, lower residues. So that's really great. But I do want to mention that, probably Max Planck people hate us for doing these comparisons, but the point for us to do that is really to benchmark the tool. We're not developing our workflow to compete on sort of on the most standard analysis. We're not interested in that. We're developing our tools because we are driven by collaborators who tell us that existing tools are not applicable or too slow or not working well for those data sets. And we use the standard analysis and comparisons with other tools just really to benchmark ourselves, because we need to know how well we are doing on standard analysis. Now, regarding speed of this type of searches, quantification, I mean, there is a tremendous improvement in the number of, in how long it takes. So that's really nice. Again, it's orders of magnitude faster than MaxQuant and a lot of other tools because of indexing at various levels in both timsTOF data as well as the Orbitrap data. Now, if you would like to see, for example, how PTM discovery, especially in the context of chemical proteomics and our label-free quantification using IonQuant, all come together in one very, very nice collaboration. And that's the type of collaboration that's really pushing us to work on this. I welcome you to go to this preprint from Stefan Hacker Lab in Germany that's really looked at. Here we looked at how we can perform proteome-wide profiling of diverse electrofields, and it's really, really nice work. Again, I welcome you. It combines everything: OpenSearch, Massive Search, IonQuant, and SciLac. Everything is combined here in one really nice application. So here is the reference. Now, I should be wrapping up, and I do have a few slides on TMT and DIA, and I'm going to try to wrap up them in the next 5 minutes, because I prepared a little last for these workflows. But FragPipe fully supports TMT analysis. In fact, we are running these workflows on so many data sets. We already, as part of the CPTAC consortium, we published it in 3 or 4 cell manuscripts now. We are running this on the pan cancer analysis. We're running this on, in other collaborations. It's working really well. It's fully functional workflow in FragPipe, but you also can run it in a sort of command line mode through the Philosopher command line option. You even can run it on the cloud through genomics data, proteomics data commons interface. So there are different ways of running this workflow on large scale in a very cost-effective way, but you could run certainly on a regular desktop, like my desktop. You can run a CPTAC cohort analysis. It would take less than a day. It's very, very fast, given the size of the data. So what are the different steps here, very briefly? So the module, TMT integrator is a module in FragPipe that will either take one PSM table, if you have just one plex, or if you have many TMT plexes, iTrack plexes. It will take a sort of a PSM table generated by FragPipe Philosopher reports for each TMT plex, and we'll combine this information. It will generate combined reports for the whole cohort. And if it's a PTM enriched data, it will generate PTM site-specific reports as well. If you have reference bridge sample, you will use that as part of the analysis. But if you don't have a bridge sample to link different plexes, you can use what we call a virtual reference approach. That sort of a reference will be created in each plex, and everything will be normalized to that virtual reference, and it works really well as well. And these reports can then go to PCA clustering analysis and downstream processing you would typically do. Now, TMT integrator has PSM filtering steps, normalization, grouping, outlier removal, additional protein level normalization at the end. All of that is implemented. And I do not have time to go into details of that. But when we show later the workflow, I hope that Sarah will be able to show some of that. As well as the reports that we generate, the gene, peptide, protein level, multi-site reports, and various important options such as using unique peptides or unique plus razor, which is the default, as well as generating tables at gene level, maybe gene level, but again, keeping only peptides mapping to one gene, as the most conservative approach, or using this sort of gene-level protein inference, and so on and so forth. An important part of that is how we generate site-level reports, where when we couple it with PTM profit, for example, for site localization for phosphodata, we also need to summarize this with respect to what sites we localized and not. So we create this site-level reports that I try to explain on this slide, but I don't have too much time now. So I'm going to move to sort of finish my presentation by saying that, again, in comparison with, say, for example, MaxQuant, on a CPTAC cohort like clear cell kidney cancer, CCRCC manuscript. So this is the publication that we have a few years ago now. We identify more proteins, and importantly, by all benchmarking, we have excellent performance of our TMT quantification strategy. For example, if we use mRNA levels as a benchmark, so we can correlate protein levels against mRNA, ask the question, what pipeline gives us protein quantification that correlates better with mRNA? Imperfect comparison, but nevertheless, one way you can compare different pipelines. So if you look at MaxQuant, we have about 8,000 pairs for which we have gene and protein abundance in this data set, and a correlation around 0.4, median mean around 0.3. That's significantly higher if we use TMT integrator, as you can see on the right. So we extensively benchmark this against other tools as well as part of the CPTAC consortium, and it's been adopted. We are now running, or rather already finished running it on 10 cancer analysis across 15 different cohorts, like clear cell CCRCC here. So it really scales to very, very large data sets and gives a really good performance based on all our benchmarks. Now, running out of time, really cannot talk about this new option that we tried, that MaxQuant sort of has a median, weighted median approach that works better in certain cases. But we do not find this particularly useful except in artificial data sets, such as spike-in protein data sets, where you have really, really high ratios of 40 to 1 and so on and so forth, because the spike-in protein is very different abundance. So based on our benchmarking comparisons, we conclude that except in cases like a data set like this, and MS2 data, for MS3 data, the interference effects are not very important. So we don't need to correct for this with a weighted median approach. For MS2, there is some value in doing this correction instead of a standard median. But when we apply it to a large-scale data sets, like CPTAC data sets, we find that overall the accuracy actually suffers. So we can better estimate the ratio, but the overall accuracy sort of suffers if we do that. So we are not using this as a default strategy, but we do have an option in TMT integrator to use, instead of aggregating PSMs using median approach to the protein level ratios, we have a weighted median as an option for such data sets. So finally, very briefly, for building spectral libraries, we have a fully sort of functional workflow for building libraries from DIA data, DDA data, or DIA and DDA. We will use DIA and part of DIA. This is a strategy that is sort of pushed and pioneered a while back now, probably before people really realized that it was a good strategy, where we proposed what's called direct DIA approach of identifying peptides directly from DIA spectra in the same manuscript. We also described a hybrid strategy of building libraries from, this is figure 1 from the manuscript, where we build a library from DIA, and we build a library from DDA, build a combined spectral library, and then go to targeted extraction using that library. So the hybrid DIA plus DDA approach. So now these strategies are implemented in FragPipe. For building spectral libraries, you can either do DIA only data, or DDA only data, or DIA and DDA, and go all the way to a spectral library file that you can load to, and this is the workflow again, that you can load the spectral library file to Diane or Spectronaut, or you could use interact files generated by FragPipe and build a library in Skyline, or you could also load the libraries into OpenSWAP. So if you look at this preprint that we published in collaboration with Marcus Ralzer lab and Vadim Demichev, where we demonstrated how beautifully this works for building libraries for TeamStuff data, where amazing results. Build libraries FragPipe based on TeamStuff, passive data, DDA data, And then, you could get 5,000 plus proteins you can quantify with Diane in 5 minutes of LC time using Diane. So it really works beautifully, and I welcome you to go and explore this if you work with passive data and would like to DIA data on timsTOF DIA passive, and would like to use our workflow in combination with Diane for that data. So in summary, I told you about the open search strategy in FragPipe for PTM analysis, the quantitative workflows, and also mentioned that we have a lot of advantages for standard workflows and other things such as HLA peptidome analysis and so on and so forth. So I'm going to stop here, acknowledge my lab again, really. And I know I'm running a little bit of time because, I always run out of time. That's just the that's the fact of life. But let's see if you have any questions now, or depending on that, we can, if you don't have many, we'll soon go to the hands-on presentation, or rather sort of a live demo that Sarah and Finchao will do, and myself as well. Or we can spend more time on discussion. So I'm going to stop here and see if you have questions and see how we should move forward here.
So we have a lot of really good questions. Maybe we can go through some of those before we start the demo. So let's see, one question is about search sensitivity between closed and open search. And they say, how do you explain that some proteins have more identified peptides in closed searching versus open searching? Based on one of the figures that was in the slides.
Yeah, so why in some, well, if it's a low abundant protein, so it only, for any protein that's decently enriched in the data set at a high abundance, we gain in a number of PSMs that we identify, because we start picking up a lot of PSMs that are modified peptides for those proteins. So the only time we would be losing something if it's a low abundant protein. So if the question refers to this Bioplex APMS data set, it could be that a few experiments there where the bait protein was really poorly enriched and had itself just a few spectral counts to begin with, a dozen or so. And it's possible that in that case, we'll be losing ATs because we're not gaining enough modified peptides, but we are losing some that unmodified tryptic peptides that you would find in closed search. So it's possible, yes. So there is, for low abundant proteins, we're more likely to start losing things when we increase the search space. And for intermediate and high abundant proteins, we'll be gaining. So on average, we gain, but not for every protein.
Another one that was answered in the chat by Finchao is, can FragPipe be used to identify peptides by non-specific proteases? And they're asking specifically about DIA data. I think this is a good one because it,
so
you sort of answered
excellent question, because we have a lot of users that coming to use FragPipe just because they really want to have take advantage of the speed of MS Fragger for non-specific searches. We have optimized workflows for HLA peptidome data, endogenous peptidome data. You can certainly build the libraries for this type of data. That works terrific. Yes, so I think for everything that's related to non-specific search, I think FragPipe is a way to go. Not only because we optimized various steps, but because just the speed, just so much faster. It allows you to search for additional PTMs, and the scoring. If you have high mass accuracy data, I think MS Fragger scoring is actually pretty good. So, I think it's a very good workflow for HLA non-specific searches and endogenous peptidome data as well. Especially endogenous peptidome, if you have to go to a length of 60 or something like this, it will take you forever to run a competitive pipeline, because you have your non-specific search and allowing really long peptides if it's an endogenous peptidome data. So, it takes maybe an hour with MS Fragger. It probably will take you, a week with some other tool to run a search like that, at least based on our collaborations. That seems to be the case.
Another question I think that is worth going over in the big group is, what is, just to recap, the difference between a closed search and an open search?
Yeah, a closed search is the one where we specify that we're only looking for peptides that either unmodified peptides, so they have no mesh, or we specify specific variable modifications that we allow, right? So oxidized methionine only, and we allow isotope errors. So when you think about this, we have MOEZ value from the experimental spectrum, say 1000 Dalton, and we have all candidate peptides in a database, and we can calculate MOEZ values for those candidate peptides. And they have to match with a very narrow tolerance. And that means that we're only identifying unmodified peptides or peptides that contain the modification that we specified, or maybe an isotope error if it's a +1 shift. But everything else, any other difference is not accounted for in the search. So, you will not find acetylated peptide because it has a 42 Dalton mass shift in a closed search if you don't specify 42 Dalton acetylation as a variable modification. But open search will find that, and any other mass shift that you may have.
So then another question that I thought would make for good discussion is someone asked or stated that it would be great if FragPipe integrates global proteome and phosphoproteome search to provide phosphosite stoichiometry or occupancy values. And they weren't sure if this is already working in FragPipe.
Well, it's a great question. So anytime you do, say, phosphoproteomics analysis or any PTM analysis in general, when you look at, say, phosphopeptide abundance data, phosphoprotein abundance data, or phosphopeptide, we typically look at the peptide level, but sometimes at the protein level. We have to normalize for the global abundance, right? So FragPipe you can generate the reports with FragPipe, but it's not something that's integrated. So those tables from the whole proteome analysis and your peptide or protein level quantification from the whole proteome data, and your, say, site level or phosphoprotein level quantification from your phospho data, those two tables still need to be merged and normalized as a post-processing step. It's not integrated. It probably would be too hard to integrate into any computational workflow. It really depends on how you want to do this analysis, and that depends on the biological question that you have.
So I think that's probably good for discussion for now. Maybe we can move on into the live demos.
Mm-hmm.
I just wanted to mention that a lot of the questions are about specific kind of technical questions about compatibility with different instruments and different workflows and things like that. And so we do have that information on the FragPipe homepage, and I can post that link in the chat in a minute, or maybe I don't have the ability to do that, but
so it is probably best to post this in the Google Doc associated with the session.
I think we have a link in there, but if not, I'll definitely add one.
Mm-hmm. All right. So then I think the next step would be for us for me to pass the screen sharing to Finchia or to you, Sarah, who is next, and then you can show some of these workflows in action. So I'm going to stop sharing.
Okay, sounds good. So yeah, I'm Finchia. I'm going to show some demonstration about the label-free quantification analysis using our pipeline. So let me share my screen. Can you see my screen?
Yes.
Cool. So basically, this is a folder. This is what the FragPipe folder looks like. In the bin subfolder, there are 3 files. For the Linux users, you can use the first one. For the Windows users, you can use the last one. So I will just double click this one. So this is what the FragPipe looks like. If you are using it for the first time, you may need to configure it a little bit because those 2 paths are blank. This is basically the path to the MS Frag and the path to the philosopher. You can simply click download, upgrade to get a MS Frag and do the same thing for the philosopher. For now, I will just specify my path in my computer. So after that, it's almost done. The Python module is not needed in most cases. I will skip this one for this demonstration. So let's go to the next tab, which is a workflow. We have many building workflows for most of the common applications such as the DIA data analysis, OpenSearch, Select data analysis, TMT data analysis, Glyco data analysis, and iTrack. For this demonstration, I will just use this LFQ-MBR, which means that we perform the label-free quantification followed by match between rounds. We select it and click load. Then we load the data. Load data is also very simple. We have these 6 rounds available. We just select them. So we can assign them to different experiments by just click the consecutive. We can also use something like control 3 treatment. The biological meaning of those data is not very relevant in this demonstration. I just show you how to do that. You can specify any structure according to your own data. Then we go to the database because we need to feed it.
Maybe you can mention that for MS stats, you also need to annotate the replicates, right?
Yes.
Oh, you did that. Okay.
Yes. So when we collect consecutive, the replicates will be like 1, 2, 3, 4, 5, 6, which is required for MS stats. In our FragPipe page, we have a detailed explanation about that. I can paste you the link if you need. So next step is specifying the FAST file. If you don't have any, you can also download it by just clicking download and specify the folder where you want to have it. After clicking this one, there will be a window showing all the options, something like, do we only include review the sequence? Do we need to add decoys, add contaminants, and the species? We generally need the decoys because it will be used in the FDR estimations. But in the demonstration, I will not click yes. I will just use the one I prepared for you, and then you also can also find it from your folder we gave you. So please remember that we do need a decoy in our FAST files. The default prefix is REV. So FragPipe will detect that half of the proteins are decoys. If your FAST file does not have any decoy, you can click edit decoys here. It can edit for you. Or if your FAST file has decoy, but the prefix is not REV, you can specify your own here, or you just click auto detect. It will detect the prefix for you. Then the next step is, there's really not much to do with those parameters because the building workflow already load all those like optimal parameters for the general usages, like the variable modifications, missed cleavages, the 2 missed cleavages. If your enzyme is not trypsin, you can specify yours from this drop-down menu, or you use custom, you change it to non-specific, and type your own site, for example, something like this here. Here I will, yeah. So if your data has some kind of enrichment, such as phosphor enrichment, acylation enrichment, you need to specify a certain variable modifications here. For example, check the phosphorations and add a K to the acylation if your data have acylation enriched. Then it's almost all set. Then let's go to the validation tab, which basically is for protein inference and the FDR estimations. Most of the cases, you don't need to worry about anything about it. We all set this parameter for you. You just needed to take a look, or maybe not. You just go to the next tab, which is for the label-free quantifications. We have this match between round checked by default, and all those parameters are like set for the high resolution MS1 data, which is normal in current date. And again, those parameters normally don't need to be changed. But if you have a good reason that your data is different from the normal, like most of the data, you can change this. For example, you reduce the minimal ions to make it more sensitive. If your data have a large deviation, over all of your protein has a large deviation, you can uncheck the normalize box, but leave it on is also okay. You can also reduce minimum scans, which is the minimum required scans for a detected feature. normally we require at least 3 MS1 scans. We can reduce it if you are using FAMES data, which normally have a fewer scans available for each ion. You can also reduce the minimal isotopes, which will make it more sensitive, but again, the overall quality will be a little bit lower. And then I will not touch the isobaric. Sarah will show you that, no PTM. And then the last step is specifying your output.
Can you increase the, can you make a little bit bigger your screen? Is there an option for you to do that? No?
It's already the maximized window. Yeah.
So in everybody's individual Zoom settings, they can change it in the view options. You should be able to zoom in. It's in the top bar.
I can make it in the center if it's better.
Well, you still need to make it full screen, I guess.
Okay, cool.
Well, that's okay, I guess. Mm-hmm.
Yeah. So the last step is specifying your result folder. You can type it or you can browse it to be, for example, I test 2. Then click run. Then it will start running.
You need to change the enzyme back, I think, because you didn't put the right enzyme, but it's okay.
Yeah, I will not wait for it to finish because I will change back to the enzyme. But basically, this is how the FQ was performed. It's pretty easy. Basically, it's just one word: load the workflow, load the data, load the database, click run. That's it. No need to worry about the parameters, especially except for the enzyme and the variable modifications. Regarding the output.
So before we talk about output, can you go quickly back to the ion quant tab?
Sure.
Because here is the options that I was describing. For example, we have match between run, it's clicked, but we have below that, we have match between runs and BR options. And there is FDR of 0.1, 0.01 for the ion, and that's what we typically apply. But the key parameters that I want to mention here that maybe you're more likely to change is this NBR minimum correlation and NBR top runs. So by default, there is 0 and 10. So 0 means that we're not applying any cutoff based on how the donor and acceptor runs are similar to each other. And 10 means that we are selecting up to 10 most similar runs to be used as donor runs for each acceptor run. So if you have a data set with 100 runs and you keep 10 here, that means that you'll be aligning up to 10. So if you want to use all runs for MBR, you'll have to increase this to a number that's larger than the number of runs, as an option. So this is the two parameters: MBR minimum correlation and MBR top runs are those that I was describing when I talked about label frequentification and some of the considerations for how to do match between runs as far as what runs to allow. So another important part here to mention is that we have several different options for peptide to protein rollup. So peptide ion quant, how do you roll up to the protein quantification, right? And we have top N approach, which is something that we've done with a DIO Empire manuscript. And it's really an extension of the top N method where we select, say, top 3 or top 6. I think by default, it's top 6 most abundant ions for each protein. And you use intensities of those ions, sum those intensities, except that the DIO Empire approach, this top N approach, is modified to also remove ions that are not observed consistently across the experiment. So if you have a luxury of selecting ions, when there are many, this approach also selects those that are most reliably detected across as many runs as possible. So anyways, but more recently we introduced max LFQ approach that performs max quant like max LFQ normalization, and that's currently set as default. And the alternative is, of course, is to generate, well, actually it's generated by default MStats file So that Finchal will probably show that, because that's the file that you will load if you want to do MS-stats analysis. Instead of this normalization settings here, you will load MS-stats file in MS-stats, and that's what you'll use.
Yeah. Yes. Thank you. So here is the result part. As Alex just mentioned, this is MS-stats, which is compatible with. You can just directly load this file to the MS-stats. It will process it from here. So in this, the data structure, the control and the replicates, the information is directly from what you put here. So whether whether experiments and the replicates put here will be reflected in the MS-stats output, and which will be taken by the MS-stats later on. And other than this file, there are several others, which to me most important one is the combined protein.tsv, which contains all the proteins identified and quantified in your data. So each row is a protein, and there are different columns for different experiments, such as, because we have 2 experiments, each experiment has 3 replicates. So we have aspirin 1, which is like treatment 1, treatment 2, and treatment 3, and control 1, control 2, and control 3. So in each group of these, we have several columns reflecting the measures of certain proteins. For example, we have the total spectral count, which is total spectral count found for this protein. And the unique spectral count is the number of the spectral count corresponding to the unique peptide of that protein. The RASA one, the RASA spectral count, which means that is the spectral count corresponding to the RASA plus unique peptide. And then this ion count. Ion count is like the detected feature by the ion count. Each feature which is correspond to like erosion profile of a peptide. So there are also the total ones, the unique ones, and the RASA ones. They are followed by 3 types of intensities: total intensity, unique intensity, and the RASA intensity, which is from all the peptides and the unique peptide and the unique plus RASA peptides. Please remember that if you click the max LFQ here, the intensity of those 3 columns will be from the max LFQ. If you click the top N here, the intensities will be from the top N algorithms. For the general usage, we recommend you using the RASA intensity for your downstream analysis. And the rest of the column are like some kind of duplicate for the different replicates. And then the last one is like indistinguishable proteins, or like mapped proteins for certain protein as a reference. And there are also other files which is useful. For example, the reprint int file can be used in the reprint website for the interproteome analysis, and the reprint of spectral count is recorded all the spectral count for each proteins, which can also be used in the reprint project, which is
right. So if you do affinity purification mass spec, you would annotate the bait protein as bait gene name in FragPipe, and you'll put control for the controls, and you would load, say, reprint SPC to reprint-apms.org for saint and fold change scoring, followed by network visualization and network reconstruction. So that would be a workflow for APMS data analysis.
Yes, thank you, Alexey. So other than those files outside, there are 6 folders, each folder for each experiment. So the structure of each folder is very similar. There are some detailed results, including the PSM level, the peptide at the protein level, and the peptide level and ion level summarizations, including the intensity and etc. things. There are also a figure, which is also showed in the Alexey's presentation, showing the FDR estimate, the model in the FDR estimation of the MBR, and also some other detailed result which can be used later on by the FragPipe to reprocess the data. So I think this is almost everything. And the last thing I would like to mention is that if you have any questions, have any errors running our tools, when you ask questions to our GitHub page, you can include this log file, which is very, very helpful for us to troubleshoot the problems. with this log file, we can basically troubleshoot like 90% of the problems. So I think this is pretty much everything. Alexey, do you have anything to add?
I would add that if you customize something in FragPipe, for example, you decide to change some settings, you could go back to the workflow page and click on save. And you'll say, you add something, dev.true or my own or customized for this experiment and save it. So next time you'll just load that workflow. It's that easy. You can even email this workflow to someone. So I email workflows to a lot of people when they contact me, and I customize it for them and email them back. So that's a very useful feature. You can find those workflows in the workflow folder of FragPipe. And if someone emails you a workflow or you download from a published paper, there are some papers already that use this as a part of the publication, or soon will be one. So you can download that workflow, place it in the workflow folder, and when you open FragPipe, it will show up in a list of workflows, and you'll load that, and you'll duplicate basically the analysis done by someone else using that workflow that you downloaded.
Yeah, thank you. So Sarah, it's yours.
I think before we move on to TMT, I think two questions in the chat that are good to discuss now. Is the MS Stats output generated as a default setting, or does it have to be specified in the software?
So if you are using you have the iron corner checked, it will be generated automatically.
Great. All right. And then the other question is there a way to generate a combined peptide output with the peptide intensities for all peptides detected across all samples?
Yes, there is. Let me show the screen very quickly. So here, can you see my screen?
So this is invalidation tab at the bottom.
Yeah, you check this one, it will have a combined peptide.tsv output. So it is what you want.
Right. In filter and report area of the validation tab, check on generate peptide, combined peptide file.
Yes.
And one more question is the NBR peptide FDR 1% common, or is that a little bit high?
Yeah, let me answer that. What to specify is the NBR FDR, right? So we recommend 1%. And if you read the paper, we have an extended discussion on that. Because in typical data sets, like, HeLa, any cell line tissues, we do not see a need to go beyond 1% because we just start adding noise. We're not transferring a lot more ions, but we start getting a lot more false transfers. Now, single cell proteomics is a different data, or sparse data sets in general. So we did find that there is a value in potentially going from 1% to say 2% ion level false discovery rate, maybe even 3% in some of the single cell data sets, if your downstream analysis can tolerate an increase in the number of false quantifications. Because the gray zone, especially if it's a famed single cell, there are a lot of really low quality ions. So you may want to increase the sensitivity of quantification by allowing some of those ions to be quantified and transferred, but they may be poorly quantified and maybe even false transfers, especially if you use a library run that's, many cells, and you transfer ideas to a single run that has very little single cell run. So that sort of still needs to be explored how to do it best, but it's a really good question. So I'd say explore going to 2%. But think about what downstream analysis you're doing and whether that analysis requires really high confidence quantification data, or it can tolerate some of the outliers and poor quantification, because you're just interested in the pathway level analysis, for example. That's okay in that case.
All right, I think I will go ahead and get started with the TMT demo. So hopefully everybody can see this. If it's still too small, one thing that you can do is go up to the top at the green bar where it says you're viewing Sarah Haynes' screen. There should be a little tab next to that says view options, and you can set that to 100%, which should make it a little bit bigger. Hopefully that works for some people. Okay, so this is the start page of FragPipe, and I'm going to show you how quickly you can load and analyze a TMT dataset. So I'm showing FragPipe here already configured, as Fengchao mentioned. If you have problems with that, or if you have questions about that, we have some information on our main FragPipe page about how to do that. And we can always answer questions on our GitHub, which is a good way to get in touch with us. So we will, I think I pretty much only have time to do the phospho TMT demo. So for this data set, we're going to use the TMT phospho bridge workflow from the workflow tab. Go ahead and select this, and then you always need to make sure that you click load to load the workflow. And this is going to set, again, all of the downstream parameters for this workflow, like automatically through all of these different steps. And then we just need to add the data next. So we've got here some 2 TMT 10 plexes, and they each, so those are 2 fractions each from 2 different plexes. So pooled samples or multiplex samples. And we will do something a little bit different for this. So we will add a folder recursively so that it will automatically load all of the files automatically. Okay, so in this case, I'm just going to select the top level TMT phospho raw folder, and that will load all 4 files. So that's, again, 2 fractions from 2 different plexes. And in this case, we wanted to label them by the name of the folder. So we're just going to use this button that labels them automatically by the parent directory. And you can see now that the plexes now have the appropriate labels given by the folder names. So that's one quick way to do that, especially if you have a really, really big experiment. That's one nice thing. And again, we recommend that you organize all your files like this, and then you have the annotation table that gives all of the sample names to each TMT or iTrack channel in that file. And so it's just a nice, easy way to organize it, and it makes it really easy to load into FragPipe. So I think I need to, and someone had a question about downloading the database as well. So I'll just go ahead and, so right now I think this is a mouse database. So I'm going to just show how easy it is to download a database, because this is a human data set from the a CP Tech data set. So I'm just going to click download and choose the folder first. And then here, since I'm using a human data set, I'm just going to leave all of these settings in the default. So this is reviewed protein sequences. We're going to add decoys and add common contaminant proteins. And then it will download. And one thing here we usually recommend that you use FragPipe to download the database, just so we can be sure that all of the FASTA headers are formatted correctly. But one way that you can kind of check that things are working well is that you do have the decoys in your database, and this should say 50% here. Okay, so we can take a quick look at the search parameters set. So we've got our precursor and fragment tolerances. We have, in this case, it's phospho-enriched data. So we've got phospho set as a variable modification. And then down here, we've got the TMT tag set as a fixed modification on lysine, as well as all the peptide N termini. And then on the quantification tab, you can see that the way that we've loaded, or actually the way that we've pre-organized the files, we've got these annotation files already inside each one of the folders. So FragPipe automatically recognizes them. And if you want to see what those annotation files look like, you can edit one of them and see here we've got the different TMT channels and the sample names, and you can edit those. And here, in this case, we're using a pooled sample, and that will be used to normalize between the different plexes. So you can see this is called pool 02, and the reference sample tag is has the pool kind of substring in it. And if you look at the other one, this is pool 01. So they both have that pool, and that's how TMT Integrator, the tool here, is able to recognize those as the reference samples. Alexey, is there anything else we wanted to note on this tab? I think one thing I'll say is that, oh, go ahead.
I don't know if you go ahead, describe this part first. Mm-hmm.
Okay, so in this case, we are not doing we're not just doing a global proteome quantification. We're interested in the TMT quantification of the specific phospho sites. So we have the modification tag here that specifies that we're indeed interested in the modification, the phospho mass additions on ST and Y. And that's how the reports will be generated.
Yeah, so this part we're actively working on streamlining this a little bit. So in the next release, it may be easier to specify modifications to TMT Integrator for what modifications to specify the reports. If now you need to know what is the mass shift sort of, and what's the total mass, which, you know, not very intuitive. Like you need to know that it's STY where it's the T181, right? So we're working on that, improving that. But what else is here? I think the most mistakes have probably come from, if someone was running something like this, is from annotating the samples. So sometimes you have an experimented TMT plex where you have two reference channels, two bridge samples. So for example, in here we have, it's a 10 plex, and the last one is a pool sample, right? And this is plex number 2, so we annotate it as a pool 2. But sometimes you may have a duplicate of that reference channel, and and people can annotate it as a, you know, with the same name in the same plex, for example, pool 1 and pool 2.1 or something like this, and pass pool as a reference sample tag. So that will create problems. You really want to have your bridge sample as a unique text name, unique text tag in this annotation file. So you would just rename the duplicate of that reference sample, you would rename it with some other word. Instead of using pool, you would pull reference, bridge, wherever, just that it would not match the one that you want to use as a reference sample for bridging plexes. That's one comment. I observed that as a common issue. Other than that, it's all pretty straightforward, I would say. Again, make sure you know your data, whether it's a TMT 10 or 11, or if you're quantifying from MS2 level, MS3 level. We are creating more workflows, for example, for real-time MS3 workflow that you can do on Eclipse. We can provide we probably will provide a separate workflow for that, a little bit customized. So things like this. So overall, it's pretty straightforward to use, again, if you stick to the basic stuff, but it's easy to customize as well.
Right. So we kind of went and looked at these things and spent some time on that, but really all you need to do is get your files organized and make the annotation file, which you can do it beforehand, or you can also create it here, and FragPipe will, once you've got the right plex set here, you can generate that file and then just create it in FragPipe if that's easier as well.
Yeah. So, if you're analyzing just a single plex, for example, or a few plexes, and it's easy to annotate, I would not create this annotation file as a text file, but just click here on Edit, Create, and if I don't even care about the sample names, I already know them by heart, I'll just keep the default name. So if you click on Edit, Create, sorry, if you click there, you will see that it will populate it well, you have to, I guess, yeah, you can select and load, and will be sample 1, 2, 3, 4, 5, right? And maybe that's all you need, really. Or you can go and manually change the names. But for large, like, you can go and change to whatever, your cell line names. If you work with, when you work with something like CPTAG, imagine that we have 23 plexes, each one has 25 fractions. Here, this is just 2 fractions for each plex because we wanted a shorter data set for tutorials. But if you have 25 files in each one, and you have 23 plexes, well, it doesn't matter how many files you have, it matters that you have 23 plexes. You're not going to go probably manually annotating all of this, right? So it's easier to create them in advance and place in each folder along with the MZML or raw files. For this version, you have to use MZML. In the future, we also, the next release will probably allow you to analyze raw files directly as well. So anyways, you place annotation files, and they need to be named the same way, annotation.txt, and placed in each folder with corresponding to that plex. So then FragPipe automatically would load that. So that's the option that we recommend for large-scale TMT analysis. It's pretty convenient once you get used to it. And another thing is that in the next release, we'll write the MStats compatible file. I don't know if you thought I mentioned that or not, but we already have that in Philosopher, a way to generate the reports. I think we just need to reshuffle some of the columns, but basically there will be an option for you to generate a MStat compatible file. So if you don't like, or in addition to running TMT integrator that has its options for PSM filtering and averaging PSM ratios to the protein ratio and so on and so forth, and outlier removal by generating MStats report, PSM table in a MStats compatible format, you can load to MStats, and I know you covered that in the course, so that will be an additional nice opportunity for you to utilize MStats as an addition to the FragPipe workflow for downstream analysis.
So I guess to quickly recap, and then I think we can go on. But anyway, so after this, after you pick your workflow, load your files, you can, of course, take a look at your search parameters and make sure they make sense for your data. But these default workflows are really what we recommend, at least to get started. And in a lot of cases, you really won't have to change anything. They should be pretty robust, and they will work. So once you load your data, pick your workflow, make sure you've got a database set up, then all you have to do is, I guess, double check your annotation files and then click run, and you will have some results. And I'll show really quick what those look like. So you'll have these, again, you'll have these reprint files, you'll have log files, and these are always helpful if you have an issue. This is where we're going to ask you to share with us on GitHub, is the log file. And there's a combined protein quantification file. And then we have individual reports for each plex, but then kind of the main thing to focus on here is the reports from TMT Integrator, which will give you reports at the phosphosite level. So there are single site, so that's like one residue in a protein that's summarized to this level. Maybe we can take a quick peek at this. It doesn't take too long to load. Oh, right, of course. To open that with Excel. Okay, maybe I don't have Excel installed on this machine. That was an oversight. But in any case, this will give you the TMT integrator summarized ratios to the single phosphosite level. And then we also have peptide and protein and gene and multi-site reports as well.
So multi-site is a report where we keep localized and unlocalized and doubly phosphorylated peptides, all these different combinations, sort of non-redundant combinations as separate rows in that file, where the single site collapses everything to the single site level information, including removal of sites that are not localized. A single site report will have only localized sites, at least localized somewhere in the data set, and at least for one PSM that was localized. And And there is also some other processing to sort of collapse this to a single site level that we'll explain in detail in the manuscript that we are preparing, or it's already explained in some of the published manuscripts.
So one question that's in the chat that is relevant is, what if you don't have a pool for the reference sample tag and all channels are used for control and treatment? And in that case, we have something called a virtual reference, which you can just specify here. And that's how you take care of that situation.
So what virtual reference is doing is that for each PSM, it will calculate in sort of an average or median of all of abundances of that PSM across all TMT channels, and it will take a ratio of each channel intensity to that average intensity. So that's, you know, when we use a reference sample, the conversion, PSM level conversion to ratio is taking ratio to the specified reference sample, right? So every TMT intensity will be recomputed as a ratio to the specified reference channel intensity. But if you don't have a reference in a data set, then you calculate the virtual reference that's average of all TMT channels and compute as a reference to that. So you can think about this as, in a way, we will be aggregating PSMs from different data sets, from different pluxes, without having a bridge channel, but assuming that kind of the average abundance of each peptide in each plux is about the same. So that's sort of the assumption for this virtual channel. And it's actually worked pretty well if your pluxes are well balanced. If one plex is all tumor samples and the other plex is all normals, you know, it's really hard to, it just, there is no way you can bridge them well, right? But if you design your plexes in a way where tumor and normals, for example, are mixed, as the case in CPTAC studies, so the virtual approach is actually working almost as well as the reference approach, but without using the reference, because the samples are mixed, and that strategy works.
Someone asked if there is a TMT6 plex option. There is. That's again just here. Let's see, another question is it possible to add your own custom isobaric tags as an annotation, or iTrack and TMT the only ones available? So right now we have the different, so these are hard coded at this point. So these are the only isobaric tags that we have available. But if you're working on something interesting, maybe you can get in touch with us and we can see what we can do for you.
I see one question that several questions, correction for label impurities. We have several questions. And also, can you enter the correction factors for TMT labels? The answer is no. Well, in fact, we implemented that correction a while back. And to our surprise, it was not helping at all. So at the moment, we actually not we don't have this ability to enter specific corrections, for each that you receive for each kit. But in our benchmarking, again, against the tools such as Protein Discover or SpectrumMio or some other tools that do have this correction, or MaxQuant, and performance was really great without that correction. So, and in several scenarios, we identified data sets where other tools were actually applying the correction that was only creating artifacts in the data, especially when it came to highly differential proteins. So the short answer is we don't have that correction, and the long answer, or complicated answer, that I'm not sure that we want to have that correction. I don't know what you think, but this is what my thinking is at the moment, at least.
Yeah, someone, I think maybe on the YouTube live stream, said that that this made a big difference for the proteome profiling experiment. So that's something that maybe we could look into and see if it's worthwhile for that.
Contact us if there are data sets where you would like to use FragPipe and you think that you really have to have that. Contact us. We can discuss that again. In our experience, it was not helping. If there are experiments where it's really necessary, we can think how to add that. But the data is, frankly, there are so many other issues in TMT data that, I don't think the isotope correction is the one that I would worry the most about. How do you remove outliers, aggregate PSMs, you get the high quality, really confident scores, the filters, and all those things affect quantification a lot. Perhaps a lot, I think a lot more than the impurity correction.
One other question, what is the difference between TMT phospho and TMT phospho bridge workflow?
Yeah, so in the TMT phospho bridge workflow, well, they're identical workflows, except in the TMT phospho bridge, what's selected in this tab we're looking now is this reference, define reference, and I think there is a bridge as a, so it's really the only difference is that in one case it's virtual and the other one is a reference with some tag. So we created it for historical reasons because we wanted to provide a workflow that will replicate pretty much the CPTAC analysis where there was a breach approach used. So we wanted to not just have a default workflow, say with virtual setup, but also the one that's for the exactly what was done for some of the analysis. So we basically duplicated the workflows, but that's the only difference I can think of.
So I don't know if I guess it's 1:56. Do we have time to, Alexey, do you want to show the DIA workflows
Well, let me see if I can quickly answer if there are any
other questions.
The aggregation method affects one experiment at a time or both. I actually don't understand that question, frankly. Maybe the person who asked Susanna can rephrase that, or someone else who understood can answer, can ask that. So the correction factors, can software handle FAMES data with different voltages? Yes. So when you do LFQ with MBR, for example, it will trace peaks for different FAMES voltages. I believe Philosopher will also trace as part of the, Philosopher has its own label-free module that's used actually in the TMT integrator workflow. I believe it will also, at least in one of the, either in the next release or it's already tracing the FAME-specific voltages. So can FragPipe build libraries from diapassive data? I mean, I can show the workflow, but the short answer is that it's not optimized for diapassive data. You could convert from .d files to MDXML files with MS Convert. But DIA-Umpire would not work well for diapassive data. And will work, but it wouldn't be great. So I would not recommend it at the moment. And I think in general, I'll say that it works so well with passive DDA data, and diapassive data is still a challenging data for untargeted sort of DIA-Umpire direct DIA approach. That I would probably recommend using diapassive data for generating the libraries for spectral libraries for downstream analysis using, say, DIAN tool. Can you also use this workflow to analyze samples with only TMT0 label? Contact us. I'm not sure if I think the answer is no, but all we would need to do is to add TMT0 option in TMT integrator, so it will extract TMT0 label, if I understand correctly. So is there a workflow for TMT labeling efficiency? I think is it a question of how to figure out whether we labeled our samples well, right? What's the TMT labeling efficiency? We don't have a workflow for that, but it's very easy to do by, our default workflow for TMT, whole proteome analysis, specifies a TMT on protein and peptide N-terminus as a variable modification. We also specify over-labeling on S, actually, for TMT, because it's very common. You could even change lysine TMT label to variable modification as well. And if you run it, you will quickly see, how many peptides you identify without label, without TMT on lysine, or without TMT on N-terminus. So you can do it that way. You could also do an open search on top of the default TMT workflow, and you'll pick up those underlabeled cases on lysine as a negative mass shift of TMT label, for example, and so on and so forth. So there are several ways how you can quickly do that, but we don't have a QC tool that will automate it for you at the moment. It's probably a good idea to add it.
But I will add that you can make that workflow pretty quickly and easily, and then save it, and you can even share it with other people. The way that we have workflows set up, so you can, if you load a custom or a default workflow and then make some changes, you can save that and then send it to other people, and they'll be able to load it into FragPipe and use it as well.
Mm-hmm. And the final one question about normalizing PTMs for this PTM abundance to the protein abundance. I think you already mentioned that right now FragPipe doesn't do it. That's something you have to do with the report yourself. Well, let us conclude by, if we don't have other questions here, and it's a lot of questions, great questions. Okay, there is a question about custom isobaric tags as notation and interact with TMT. You have to mail us and you have to add it to Philosopher, because we need to tell a Philosopher which one to extract. No, it's not possible for you to add custom tags. You have to mail us, and it's not a big deal for us to add and put it as another option. The TMT 16, TMT 26, 128, whatever is going to be there. Like a treehouse, 13th floor treehouse book series. You can go up and up and up. And every year you add one more book to the list. Now, DIA, Sarah, do you want to show quickly here? Do you have that?
Yeah, we can do that.
Again, with DIA building spectral libraries, it's so easy. So you have several workflows. From DDA data, just plain DDA data, click on SpecLib workflows and load DDA files and click go. That's it. It will go all the way to the end and will build a library. So if you load this workflow, you don't need to annotate experiments. You just load all files as with one list. Don't annotate them through experiments or replicates. Just load the workflow and build a library. If you go to the spectral library tab here, if you click on that spectral library tab, you see that RT alignment will be done using this common reference peptides, CIRT peptides, which is about 5,000 commonly observed human peptides. Now, there is another option to change to IRT, but a better one, if you're building the library from fractionated DDA data, you cannot use one of the runs as a reference run. So I recommend using this CIRT option, in which case all runs will be aligned to this extended sort of reference set of commonly observed peptides. If it's a non-human data, because the CIRT set is for human data only, or some yeast data maybe, but for other organisms, you have to have spiked in IRT peptides in a sample, and then you can select that, and alignment will be done using those IRT peptides. Now, and you just click run, and that's it. If you are building library from DIA data, direct DIA, that's where you'll select DIA umpire_SpecLib workflow and load that workflow. And then you select your DIA raw files, so MDML files. And again, you don't need to change anything. Everything is set for you already. You could, if you go to the SpecLib tab, you will see that here it will be automatic selection of a reference run, because we assume that in this case you have single shot DIA data. So instead of aligning to some IRT reference or common peptides, the preset option here is that one of the runs will be selected as reference, and all other runs will be aligned to that run in RT in retention time. Or if it's a if it's a timestamp passive data, ion mobility and retention times will be aligned to that reference run. And then you just click run, and that's it. And you'll get direct DIA analysis, and you'll build the library and just load that library, that TSV file, to say Diane or Spectronaut, or use interact files, Skyline, and that's it. Now, if you want to build combined DIA and DDA data, it's a two-step process. First, you will run DIA umpire only workflow without building the library. So you can click on that, and it will not build the library. It will only run the DIA umpire step. And here you have some options that you can actually, we can take a look now, and you can use default options, and there are instructions for how to change them. If you don't mind, longer computational time, you can set some options to, as recommended, to high sensitivity. If you want to run as fast as possible, there is another option, and the default is sort of the optimized analysis. So if you would like to build a hybrid library, and after that, I think we'll conclude. But if you want to build a hybrid library, you will run DIA umpire only workflow that will only run DIA umpire to extract pseudo-maximal spectra from DIA data. And they will be in the folder where you write the results that you'll specify. So you'll have to then continue and load spectral library workflow. And we have it all explained as a tutorial on the website. You'll load spectral library workflow. And now in this workflow, you will load both DDA files and DIA umpire extracted pseudo-maximal spectra MDML files. So you'll load from two different folders. From your DDA folder, you load DDA data. From your results folder for the DIA umpire run, you'll load MZML files created by DIA umpire. And then you analyze all the data together through the spectral library workflow all the way to the end. And you'll build a combined library. That's very, very simple. It's as simple as that. So results are really great. You compare it with Spectronaut, they're pretty much as good as Spectronaut. Spectronaut implemented the umpire workflow as part of that tool. We have really great results now with the umpire in this in FragPipe, especially if you combine the data with some DDA data for hybrid library. We're also working on additional workflows, so there is a lot more will be coming. There is MS-Fragger DIA workflow. I don't want to go into that workflow right now, but there is an alternative workflow to DIA umpire with direct DIA that's already available. If you're into DIA analysis, you can contact us. We can explain that, or we'll explain it better in the near future in a manuscript and in a tutorial.
So we have another great question in the chat, which is, if I want to analyze the IDs from FragPipe or with Skyline later, how do you suggest the use of a spectral library generated from the interact.pep XML outputs, or can FragPipe generate a compatible library output?
Yeah, so that's a great question. So actually, what I've done so far is loading interact files, and without going through all the way through building the spectral library, just taking basically peptide profit output files and loading them into Skyline interact with PepXML files, and it will find MZML files and will build a library in Skyline. It works great. Now, I also recently tried loading library.tsv file in Skyline, and for some reason it gave us an error. We have to follow up with the Skyline team to see what in a library.tsv file that is not recognized by Skyline, because this library.tsv file generated by EasyPQP FragPipe, we can load into Spectronaut, we can load it into Diane tool just fine. So I'm not sure what Skyline is missing, or rather what our library is missing, but we'll figure it out. And I think we should have an option to allow loading library.tsv file into Skyline, because it may be a beneficial option, since our libraries are filtered based on protein FDR and peptide FDR. They're protein inference sort of analyzed. I think they're really good libraries. So I think it's a good option for Skyline users as well to compare building library inside Skyline from the search results versus taking the library that we have, because we also have nonlinear retention time alignment already done in there. If it's the DIA passive data, we have eye mobility aligned as well. So it's something that we'll look into in the future.
So we're 10 minutes over now. I don't know if we can stay on and keep answering questions.